end replication problem in eukaryotes. Actually, what is it? What happens in end replication problem? I already told you DNA molecule in eukaryotes are linear, while replicating linear double-stranded DNA, the 5' end of each new DNA strand will generally commence greater than greater than 100 base pairs short of the 5' end of the template strand. Did you understand that word? Little bit of complicated. Okay. DNA molecule of eukaryotes are linear. Just imagine that eukaryotic linear double-stranded DNA. Okay.
Yes.
The first strand is template strand. The lower strand is the template strand. The upper strand is the newly synthesized daughter strand. Okay. Yes. Okay. While replicating linear double-stranded DNA, the 5' end of while replicating linear double-stranded DNA, the 5' end of each new DNA strand will generally commence greater than 100 base pairs short of the 5' end of the template strand. That means the length of the daughter strand, the last part at the time of replication, we know we have to terminate this replication process. Yes or no? You have to finish this replication process. So the last part of replication, what is the difference between template strand and daughter strand? Okay. Okay. So the 5' end of each new DNA strand will generally commence greater than 100 base pair short of the 5' end of the template strand. That we will discuss in detail. We can see that what is that mechanism. Okay. Each time cell replicates its genome, in order to divide, its chromosome becomes shorter. Okay. Chromosome becomes shorter and shorter because of this problem. This difficulty is resolved by telomerase replication. I know it is not clear for Sandhya. Yes? Is it clear? This slide is not clear for you. Okay. The last time of this class, it will be clear. Okay.
Yes. Okay.
Okay. There is a length problem between this parent strand and daughter strand. What happens in that case is the parent strand is length of parent strand is different from daughter strand at the time of termination. You just understand that point. Okay.
Yes, ma'am.
At the end of replication, the length of parent strand is different from daughter strand.
Yes.
Okay? The mechanism of how it is different. What happens when it is different? That we will discuss later. Okay. So this difficulty is resolved by that particular process is known as telomerase replication. Okay. Okay. The next slide. Next slide. Yes. Telomerase replication in eukaryotes. Before entering into telomerase replication in eukaryotes, we must know the general idea of replication. We know what happens. Replication bubble is there. Then leading strand synthesis with one primer. Lagging strand synthesis with numerous primer. One helicase is there. You can see this helicase. Yes?
Yes.
Okay. You can see this helicase. Then helicase moves and this DNA strand unwinds. Okay. Okay. You can see here helicase along with MCM 227. What is this MCM 227?
Mini
Yesterday. Yeah. Mini chromosome maintenance protein. That is hexamer. It's a hexamer that contains 6 subunits. Yes. 2 to 7. That helicase is loaded along with this MCM. Okay. This helicase moves to this end. This is the last portion. Consider this is the last portion of replication. Okay. So the last replication bubble. So helicase moves to this side. Then what happens? The DNA unwind. Here the leading strand synthesis progresses. Here the lagging strand synthesis progresses. Here only one primer. In case of lagging strand, more than one primer is there. The primer, which direction? Primer synthesis in which direction? 5' to 3' direction. Direction is very important. So stick on that direction. Here the leading strand synthesis that occurs in 5' to 3' direction. Here the template strand that run in 3' to 5' direction. We have to synthesize in 5' to 3' direction. That's why in reverse direction. So the DNA replication is known as bidirectional. Is it clear? First class we discussed DNA replication is bidirectional. Why it is bidirectional, you know. Okay, then helicases moves to the end of the DNA, it unwinds. So the next slide, we can see what happens when the helicase come to the end portion. Next slide. Yes. So in case of DNA replication, DNA molecule unwanted to replication bubble by DNA helicase with MCM, leading and lagging strand synthesis occurs. Helicase moves to the end of the strands. DNA strands are separated at the end region after the removal of helicase. Continuous synthesis occurs in leading strand with one primer. Discontinuous synthesis occurs in lagging strand with more than one primer. Okay, so I think you are clear. Now we are in the end of replication. So there is only one replication bubble. Okay, we have two strands. Normally replication carried out. So what is the difference that we will see later? Okay, the next slide. We can see what happens. Now the helicase moves to that is the important point. Helicase moves to the end of the strands. Then the strands are separated. Okay. Next slide, we can see how the strands are separated and what happens when helicase is removed. The next slide. Yes. This is the end portion of double-stranded DNA strand. Clear?
Yes, ma'am.
Yes or no? Yes. Yes. What happens is that replication bubble is there, and what happens? Replication continues. This helicase moves out. Helicase is not there. Unwinds and removed. Now the final part of replication is like this. The leading strand synthesis is there. Then here it is one primer is there. And then leading strand synthesis occurs continuously in 5' to 3' direction. Here we need primer, first, second and third primer for the synthesis of lagging strand. Okay, then next slide. This slide is clear. You can see what happens is there. Now replication bubble opens, helicase removed. Okay. These are the 2 points. Replication bubble opens because it is the last part of replication. Last part of DNA that coming under replication. Okay. End portion. So the end area. Okay. The next slide. We can see more in next slide. Next. Yes. So what happens in lagging strand? Okazaki fragments are formed with the help of 3 primers in the end region of lagging strand, and they are also removed. Primer synthesis occurs and these primers are removed after the formation of Okazaki fragments. We know primers are removed. Yes or no?
Yes, ma'am.
Yes, Anthea?
Yes.
Primer is for the synthesis, starting material of replication. After the synthesis of Okazaki fragments and also the leading strand, the primer is removed, and the primer removed portion is filled by DNA polymerase. Okay? Yes, DNA polymerase. Then nick joining is done by DNA ligase. We know that in case of Okazaki fragments, DNA polymerase synthesizes and there is a nick, and the joining of nick is done by DNA ligase. Yes. Here, now we discuss human replication, human DNA replications. That's why eukaryotic DNA replication. So in that cases, primer removal is done by the last slide. Primer removal is done by
RNase and FEN.
RNase H and
FEN1.
FEN1. Okay. So what happens after the removal of primer? that incorporate deoxyribonucleotides in the gap instead of ribonucleotides. Now concentrate on lagging strand because in this particular strand end replication problem occurs. Now we are concentrating lagging strand. Okay, from the diagram it will be clear. Okay,
Yes.
up to this point, can you see the points one by one? Sandhya, what do you
Yes.
understand in this class?
The
Can you please explain?
Yes, ma'am. Telomerase replication is the final part of the replication. After every replication, there is shortening of chromosome, and that also causes aging. The DNA replication inhibitors are drugs and some antibiotics. The telomerase replication. The daughter strand and the template strand are different that at the end of replication then
Okay, then now you just explain the last part. What happens in last part?
in
You
last
just-
part replication bubble is opened, then helicase is removed.
Yes,
Then
helicase is removed. Then
after the formation of leading strand, there is three primers three RNA primers formed. After that, Okazaki fragments are formed and RNA is removed. RNA primer is removed by RNAs H and FEN1.
okay, primer removal is done by RNase H and FEN1. Okay,
Yes.
then deoxyribonucleotides in the gap are incorporated instead of ribonucleotides. Now we are concentrating only on lagging strand. There is no problem with leading strand. Okay,
Yes.
leading strand is okay. Leading strand, there is no problem. It's okay.
Yes.
So what happens in lagging strand? If there is some problem with lagging strand, what happens in that lagging strand? That problem is known as end replication problem. Okay. Okay, next slide. Next slide. Yes, you can see this. This is the leading strand. Okay, leading strand. Then primers are added. What is the direction of this primer? 5 prime to 3 prime. So we just avoid that leading strand. So there is no problem with leading strand. Same length, 5 prime to 3 prime direction. The next one is in the anti-parallel strands. We synthesize also in 5 prime to 3 prime direction. Only one primer is there. Primer is removed and that primer gap is filled by deoxyribonucleotides and joined by ligase. Everything is normal. Okay. What happens in lagging strand? Now we are concentrate in lagging strand. The template strand in which direction? Template strand, Sandhya?
3 prime to 4 prime.
3 prime to 5 prime. What is the direction of primer?
5 prime to 3 prime.
So in this area, this particular portion, the end portion of DNA, we need 3 primers. Okay, 3
Yes.
primers. Okay, the primer in which direction? 5 prime to 3 prime direction. What is the importance of this 3 prime direction? That 3 prime direction contain one hydroxyl group. To that hydroxyl group, addition occurs. Deoxyribonucleotides are added to that hydroxyl group. So we need, for the action of polymerase, we need this 3 prime OH group.
Yes.
Clear?
Ma'am.
Yes?
Ma'am. Yes. Ma'am.
Okay. So that's the point. That is the important point. 5 prime to 3 prime direction. Primer contain 3 prime OH group. So here there are 3 primers. Okay.
Yes.
That's all. 3 prime to that 3 prime OH group, the addition of deoxyribonucleotides occurs. That's
Yes.
all. That's all about that. That's about this slide. Okay. The next
Yes.
slide. Next slide. Yes. Now we can see, once again explain, you can see there are 3 primers. Okay. 3
Yes.
primers. This is lagging strand and this is Okazaki fragments are there. Okazaki fragments and RNase H plus FEN1 that removes this RNA primer. Okay.
Yes.
Clear?
Yes, ma'am.
What is the direction of RNA primer?
5 prime to 3 prime.
I repeat and repeat because it is very important. 5 prime to 3 prime direction. We have 3 Okazaki fragments and 3 primers here. Okay.
Yes.
Thank you. Okay. Next slide. Next is the interaction time. Okay. That's all. Here, when RNA primer is removed, RNase H and Fen1 remove this RNA primer. That creates a gap.
Yes.
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 Here
Yes,
we can see 3 gaps. Okay.
Yes.
So here 5 prime to 3 prime synthesis occurs. 5 prime to 3 prime. Here one OH group is there. 5 prime to 3 prime. 3 prime OH group is there. So we can fill this gap. Here what is the direction? Here what is the direction? It is
3 prime to 5 prime.
no.
5 prime
5
to
prime. This is 5 prime. Okay.
Yes.
Clear?
Yes.
Concentrate. Okay. The first one. Okay. There are 3 gaps. Primer gap 1, 2, 3. 3 primer gap is there. So the first primer gap, that here Okazaki fragments synthesis occurs. First Okazaki fragments 5 prime to 3 prime, then 5 prime to 3 prime. So here the end is 3 prime. Here the end is 3 prime. What about this end? This end is Sandhya? This end is?
I cannot see where you are pointing with this thing.
Okay.
That is end, which end I do not understand.
Okay. You cannot see the arrow like. Okay.
Yes.
Okay. So there are 2 strands, leading strand and lagging strand. Now we concentrate on lagging strand. Okay.
Yes.
Okay. Then you can see RNase H plus Fen1. 3 arrows are there. Yes. 1, 2, 3. 3 arrows. Okay. Now you can see the point.
Yes, ma'am.
Okay. 1, 2, 3. 3 arrows are there. Okay. So what is the 3 arrows that indicates?
The gap.
Gaps. Very good. Primer gaps. Okay. That indicates the primer gap. So first gap, second gap, third gap. So after first gap, you can see 1 Okazaki fragments. Which is that direction? Which is the direction? 5 prime to 3 prime. Then second Okazaki fragments. In which direction?
5 prime to 3 prime.
3 prime direction. So in the second gap and third gap, we have 3 prime OH group. Yes or no? Yes. What about the first gap? That is 5 prime. Yes.
Ma'am.
We cannot add deoxyribonucleotides into that gap. Yes or no?
Yes, ma'am.
Yes. So that a particular portion is free. What happens to that area? You can see from the second diagram. I already drew this for the lagging strand, parent strand 3 prime to 5 prime direction, and newly synthesized strand. There are 2 3 prime OH group. 3 prime OH group. Because of that 3 prime OH group, polymerase alpha can synthesize RNA primer gap. Yes. In between those 2 gaps, it is those 2 gaps are filled by polymerase alpha. Clear?
Yes, ma'am.
What happens? Question mark. What happens to this first gap? Yes?
Yes, ma'am. Understood.
Now, what about the length of daughter strand and parent strand?
Daughter strand is smaller than parent strand.
Clear?
Yes, ma'am.
So, I already told you there is a problem, end replication problem, that is between the length of the daughter strand and parent strand. Okay.
Yes.
Okay. So, here the daughter strand, last primer is removed. In this case, it's first primer, but actually it is last primer. Last primer is removed. In this area, what happens is that we cannot fill that gap with DNA polymerase because of this 5 prime group. We need a 3 prime OH group. Yes? Then
Yes, ma'am.
only we can fill the gap with deoxyribonucleotides. All polymerases add, right here, all polymerases add nucleotides at 3' terminus end, hydroxyl end. Which
Yes,
polymer? Polymerase alpha.
polymerase alpha.
We know, last class we discussed about alpha, beta, gamma, delta, epsilon.
Yes, ma'am.
Alpha, that is the initiator polymerase that contains primer activity and also polymerase activity. Just starting synthesis, okay? Yes?
Yes, ma'am. Yes, ma'am.
That have no proofreading activity like that, okay?
Uh-huh.
So,
Yes, ma'am.
yes, so here is the problem. Okay, next slide. I think you know the problem. Okay, so how can we remove, how can we correct this problem? Okay, how can we correct this? If we are not correcting this problem, what happens? The length of DNA reduces.
Reduces. Yes, ma'am.
Okay, actually the parent DNA contains some genetic material. It will not be copied. Yes,
Yes. Yes, ma'am.
that will lost. Our important part will lost. Yes.
Yeah, ma'am. Yes,
That
ma'am.
will lead to some problems will create that again and again. What happens? That will lead to cancer like problems.
Yes, ma'am.
Okay,
Yes.
so we have to clear that problem. That problem is cleared by this enzyme. That particular enzyme is known as telomerase.
Yes, ma'am.
Yes.
Yes, ma'am.
Okay, I think you are clear with what is this end replication problem. It is also known as telomerase replication, but actually what is the end replication problem? Now you understood.
Yes.
Yes?
Ma'am. Yes.
Okay. Okay, then next. Yes, so we just revise. After primer removal, we get lagging strand with primer gap, which is need to be filled by DNA polymerase. All polymerases adds nucleotides at 3' OH end. Here two gaps are. Yes, Sandhya, any doubt?
No, ma'am. Understood.
Are you follow me?
Yes, ma'am.
Can you follow me? Yes.
Yes.
Here two gaps, only two gaps are filled by DNA polymerase alpha. One gap has remained there. There is a problem at last, primer gap, because in that gap, what happens?
There is no 3' OH group.
There is no 3' OH. We must understand the structure of DNA. It is very, very important. I think you know the structure of DNA.
Yes.
Yes. Each and every part of DNA. That is very important. Then only we can easily communicate 3' OH group, 5' position, 3' positions. And what is this 3' OH? Okay, that is in the, just read the structure of DNA. Okay. Okay.
Yes.
Then there is no 3' OH in case of first primer gap. That in that portion we have only 5' end.
Yes.
Okay. Last primer removal that creates this problem is solved by what particular process? Which is that process?
Telomerase replication.
Okay, very good. Telomerase replication. Now we are entering into telomerase replication. Okay.
Yes.
Next slide. Next slide. Yes. Most cases, the hanging part of parent strand is removed by FEN1. In most cases, okay? Okay,
Yes,
Sandhya?
Yes,
in most cases, hanging part of parent strand is removed by FEN1. Just 1 minute, wait.
Yes.
Hello? Okay, Sandhya?
Yes.
Okay.
Yes.
Most of the cases, what happens is that hanging part of parent strand is removed by FEN1. Okay?
Yes.
Then what happens? Hanging part is removed. That is the main part of genome. This is a part of genome. We lose some important information. Okay? Every cycle, this process repeats and repeats. That shorten the length because of the end replication problem. This is the reason for aging also. This is one of the reason for the aging. Okay.
Yes.
Okay. Some cells overcome this problem with this telomerase enzyme. Telomerase enzyme, we already discussed telomerase enzyme that has got own RNA template and polymerase activity.
Yes.
Okay. Then one speciality is there. telomerase enzyme that work only on parent strand. Okay?
Yes.
It can act only on
Parent.
strand. The telomerase is mainly work to conserve that parent strand.
Yes.
Okay? It want to conserve that parent strand, conserve the part of genome without losing,
Yes.
without removed by FEN1. Okay?
Yes.
FEN1. Okay? If it is hanged, FEN1 will remove that portion. Okay?
Yes.
So
Yes.
we are avoid that situation by the action of telomerase. We avoid that situation. That's why it is that works only on parent strand. So first we concentrate on lagging strand. Yes, lagging strand. Now we concentrate only on parent strand. Okay. Yes. That elongate. What happens to that parent strand? That elongate parent strand at 3' end. What is the role of telomerase? Actually, that telomerase enzyme bind to parent strand and elongate that parent strand. In which direction parent strand run? Which direction?
3' to 5' direction.
3' to 5' direction. So the same direction, 3' end, the length of the parent strand increases. Okay? Yes. That is the role of telomerase. We can see from the picture diagram. Okay, next slide. We can see in detail what happens by the action of telomerase. What happens, we can see from the diagram. Okay. Next slide. Yes. Yes?
Yes.
You can see telomerase replication. Question mark part, the last primer gap. It is cleared by telomerase replication. That can be removed. That part can be removed by FEN1, but it is a part of genome that contain important genes. Yes? Okay. Next slide. Next slide. Next slide. Just wait. Yes. Now we can see from the first diagram, first starting point, the extent portion, the hanging portion of parent strand. Okay, black color strand, parent strand. One hanging part is there. That hanging part is part of genome, so it contains important genes. Okay.
Yes, ma'am.
Okay, Sandhya. Okay.
Yes.
So what happens? We have to conserve that hanging part. So for that, we need to add deoxynucleotides in that missing portion of daughter strand. Okay, but we have only 5' terminus end. That is the problem. Yes.
Yes.
Reading, right in Reading needs to add deoxyribonucleotides, but we have only 5' terminal end. So we can't add deoxyribonucleotides. How can we clear this problem? With the ends of with the helps of telomerase enzyme. Telomerase enzyme that contain one RNA template. One RNA template is there with telomerase enzyme.
Yes.
Okay. Yellow color, that is known as telomerase enzyme that contains CAA, CCCCA, that is RNA template. Okay.
Yes.
Any doubt?
No, ma'am. I understand.
Okay. Next. You can see this telomerase enzyme, lower part parent strand that contain telomerase sequence. Now we don't have, so the first slide. Now we don't have, yes, this diagram that explains telomerase enzyme. The telomerase enzyme enter into the hanging part, okay, and attached to that hanging part, okay, attached to that hanging part, and elongation by telomerase occurs. Which strand elongate? Which strand elongate?
Parent strand.
Only parent strand.
Yes.
Okay. Only parent strand elongated by this enzyme telomerase. In which direction that elongate? 3'
end of the parent strand.
Okay. That direction 3' end elongates. Okay. From right to left, that enzyme moves, and 3' direction that strand elongates. Okay. Then next slide. Okay. Lower parent strand contain telomerase sequence. The next slide, we can see the telomerase sequence GTT, GGGG, TTGG. Okay.
Yes.
Now we don't have 3' end for the action of polymerase. Here comes telomerase enzyme. Telomerase is known as a reverse transcriptase enzyme that carrying the RNA template. Last two bases of telomerase enzyme bind with last two bases of the parent DNA strand. DNA synthesis occurs and parent strand elongates towards the right. The actual parent DNA occurs. That we can see from the diagram. If you don't know anything. So the lower parent strand contain one sequence. You just imagine the sequence is there. Which is that sequence? Can you say that sequence?
GTT, GGGG, TTGG.
Okay, that's it. Just understand what is telomere sequence. Now we don't have 3' end for the action of polymerase. Here comes telomerase enzyme. It's a reverse transcriptase enzyme that carrying RNA template. Okay. In which point the telomerase enzyme bind? Okay?
Yes, ma'am.
Yes. The last two bases of telomerase enzyme, okay?
Yes, ma'am.
That contain an RNA template. Yes or no?
Yes, ma'am.
Telomerase enzyme itself contain an RNA template. Yes. That RNA template, the last two bases of telomerase enzyme bind with last two bases of parent DNA strand. Telomerase sequence is there. GTT, GGGG, TTGG. Okay.
Yes.
Okay. So last two bases of telomerase enzyme bind with last two bases of parent DNA strand. DNA synthesis occurs and parent strand elongates. Yes?
Yes.
So parent strand elongates towards left. Left. Okay.
Yes.
The right side that contains the original part of genome.
Yes.
Okay. Extra prepared genome, extra prepared part is left side. The original part is in the right side.
Right.
Clear?
Yes.
Okay.